So, welcome to the Science Salon with David Quammen. His new book is The Tangled Web. No, sorry, The Tangled Tree. Although we're going to end up talking about webs. The Tangled Tree: A Radical New History of Life. It's published by Simon & Schuster. Comes out in a couple of weeks. We'll post this the week that the book is published. And you are, to give you a proper introduction, although this will all appear in print on the website. The author of 15 books, including The Song of Dodo, The Reluctant Mr. Darwin, and Spillover, a finalist for the National Book Critics Circle Award. You've written for Harper's, The Atlantic, Rolling Stone, the New York Times Book Review, Outside, and Powder, among other magazines, and you're a contributing writer for National Geographic, and you've written an entire issue of National Geographic, May 2016, first time they've ever done this. I didn't even realize that was a thing you could do. That's pretty cool. And you live in Bozeman, Montana, with a couple of Russian wolfhounds and a cross-eyed cat, and apparently somewhere in the background there a python.
Boots the python is hiding in the bookshelves over there now.
Oh, you let him run. You let him roam free.
I let him roam free in the office, and then I have to find him. Yeah,
I read that one of your blogs talked about you adopted the python as a pet because somebody adopted it, then it got too big and they were moving or something.
Their teenage sons were leaving the house, and the parents did not want them to leave behind. So this snake was going to be out on the street, which is not good for a python.
No.
We adopted him.
Introduced species like that can cause havoc. I don't know how well you do in Montana in the winter, but
it would not be a concern that he would populate Montana. That's a terrible problem in the Everglades.
Right.
So you're right.
Right. Well, one of the things I always liked about your writing is that you're one of those people that goes out in the field. You actually get out there and do stuff and see things and travel, and it's kind of in the 19th century genre of Alfred Russel Wallace and his Malay Archipelago travel narrative, full of adventures and all that.
That is, a large part of my professional life has been following biologists through jungles, sometimes through swamps and up and down mountains and things like that. But yeah, I love being out in the field. I do a lot of that sort of thing for National Geographic, and have done that in past books. And this book is kind of a departure. One of the trickiest things about doing this book is that I'm not following biologists through any jungle.
Right. You shifted from big things, animals with four legs and trees and forests and ecosystems, to microbial life. But we'll get to that in a minute. Just since we're on that subject, I wrote a biography of Alfred Russel Wallace. He was the subject of my doctoral dissertation, and reading his adventures in the 1830s, 40s, and 50s, what it was like to go out in the field. They don't have Gore-Tex and fancy boots and Camelback water carriers and things like that. The ruggedness of these individuals. Of course, Darwin as well in the Galapagos. It's just stunning.
Alfred Wallace was a great hero to me too, and he's a main character in my book about evolution and extinction, The Song of the Dodo. That was sort of a starting point for me. He was, as I say in the book, the man who knew islands, and you know that as well as anybody, having done a dissertation on him. But yeah, what was it? 5 years in the Amazon, and then 8 years in the Malay Archipelago.
Yeah. When Frank Sulloway and I went to the Galapagos, and he's been many times. I went with just one of his trips to retrace Darwin's footsteps just on one island, San Cristobal, and it was brutally hard. You have to carry your own water. There's virtually no water on most parts of the islands, and particularly where Darwin was. And I would just be collapsed into my tent at the end of the day, just wasted. And parenthetically, this is why it took him until he got home and opened that notebook that you write about to start his theoretical speculations. He's just too damn tired at the end of the day to think big theoretical thoughts about what he had seen.
Yeah. And he worked hard. 4 years, 9 months, and 5 days, the Beagle voyage, and in exchange for that, he got 5 very, very suggestive weeks in the Galapagos.
Yeah.
And he saw some other interesting suggestive things.
One of the entries in his log was something like what I just described, where Frank and I collapsed at the end of a day. He described it as a long walk. Sure.
Yeah. Darwin was a tough field man, a wonderful tough field man when he was young. And then, as you know, he got back to England and never left Great Britain again.
Yeah, we always see pictures, and students are always shown pictures of these, great elder statesmen in their, 70s and 80s, but they did all this stuff in their 20s when they were young and strong and super fit. So as something of a departure from your previous work, I thought I'd enter this subject with this personal story. I learned evolution from when I started graduate school. I was in an experimental psychology graduate program, but just for fun, I took a course on Tuesday evenings from 7 to 10 from Bayard Bradstrom at Cal State Fullerton. He was a herpetologist. And, I knew nothing about evolution, and I had been a Christian. I went to Pepperdine University, and they didn't teach creationism, but I thought, well, that must be the dogma because I'm a Christian, and turns out that was wrong. And I remember sitting in Bayard's class thinking, my God, this evolution stuff is really true. He was just piling on all the evidence at the time, and he made us memorize Ernst Mayer's definition of species. A species is a group of actually or potentially interbreeding natural populations reproductively isolated from other such populations. I still have it memorized to this day. And but the idea of the branching tree is that that reproductive isolation is the key to speciation, according to Mayer, such that once they're split, they can't go back. And right. So let's say in his theory of allopatric speciation, I'll just describe it briefly and then we'll enter with the idea of trees with your book, is that let's say a mountain range erupts or a river flows through and divides a population, or a housing tract or a city or whatever, and a large population is split off, and maybe you have a peripheral isolate, just a tiny number of individuals over here. And over the course of millions of years, they diverge genetically because the environments are ever so slightly different. And then maybe millions of years later, they come back together when the mountain range erodes or the river is buried or whatever. And now they can no longer interbreed because they're genetically too different to interbreed. So, as Mayer says, this is how new species are formed. Just by definition, they can't interbreed, even if they look a lot like each other. And in Bayard Bradstrom's example, he was a herpetologist, so he studied lizard behavior, and he found in a species called California encetinus that these lizards different species had different bobbing behavior for mating. For attracting mates. So the one lizard species may bob 3 times and then pause and then bob 2 times, and the other one is the female's looking for 4 bobs and 2 bobs, and so they never get together. Whatever the isolating mechanism is, they can't come back together, and therefore the tree kind of makes sense. Like the branches are going off, and never the twain shall meet again, and that's the dogma you're addressing in this book.
That is one. Yes, absolutely. That is one of the sort of canonical ideas, the categorical beliefs that came down to us from Darwin and from neo-Darwinism of the 20th century, of which Ernst Mayr was one of the founders. The modern synthesis of Darwinian theory. One of the canonical understandings was that in the course of the diversification of life, lineages branch apart and never reconverge. And a species is a discrete, unitary thing. It's a population of animals, as you say in Mayr's famous definition, but it is separate, and it stays separate. But even Mayr, you probably remember this, even Mayr in his great book, Systematics and the Origin of Species from 1942, points out that there are occasional cases of hybridism, where 2 very closely related species may interbreed in places where the populations are contiguous, and they overlap a little bit, and there's some interbreeding. And it's even possible that interbreeding can produce offspring, possibly even fertile offspring. And that's one of the reasons why Mayr was concerned to create a definition of species, actual or potentially interbreeding individuals. I haven't memorized the definition as you do have, but I know that definition from his book. That's one of the reasons it was important to define what is a species, because scientists were discovering that, well, these are separate populations, and yet there is a gray zone sometimes between 2 closely related species. So there's a little bit of overlap, there's hybridism. But my character, Carl Woese, and we'll get to him, I'm sure, he comes along and he triggers this revolution in phylogenetics, in the determination of what creatures are related to what other creatures, and what has the history of life been shaped like. And his methodology and the people who followed him discover that there is movement between different branches on the tree of life, and not just in closely related species. It's not just hybridism between closely related species, but in fact, wow, gee, who knew? Genes in some cases are moving sideways from one completely different major limb into another major limb. And that was one of the challenges that Carl Woese and his followers threw up to orthodox Darwinian theory. Having said that, I am a faithful Darwinian, and as are you. Every time somebody says, "Well, there's been an emendation to Charles Darwin's theory," the creationists come running in and say, "Well, see, these guys don't even agree. It's wrong, and they're disproving Darwin." No, we're still modifying Darwin just the way Einstein modified Isaac Newton.
Right. It's not displacing the theory, it's building on it.
That's right.
Yeah.
Absolutely right.
Yeah. So, I mean, the most famous tree of all, because the first tree of all for evolutionary biologists is, of course, Darwin's. It was a fold-out in The Origin of Species. And, and it's an interesting idea to think about the role of metaphor in science, particularly with subjects we can't really get our minds around. Dawkins calls this middle land. We evolved in the savannas of Africa, where we're used to seeing middling-sized things like animals, and from ants to, say, mountain ranges is about the spectrum of our scale of our size. And for speed, from walking to running, and that's about it. So things like the speed of light, the expansion of the universe, quantum physics, none of this has anything to hook into our brain to understand intuitively. And of course, time is the same problem. We're used to thinking of things in terms of days, weeks, months, years, maybe decades at most. And most of the kinds of things we talk about, like with climate change, in 1,000 years, it's going to look like this. 1,000 years, I'm just thinking about next week. It was cold yesterday. What do you mean global warming? This kind of thing. It's counterintuitive, and evolution, I think, has always had that problem. When you're talking about species changing over tens of thousands, hundreds of thousands, or millions of years, there's nothing in the human cognition to hook that onto. Go, yeah, I know what that's like. So you have to create some transfer mechanism to get it from here to there. So the tree has always been that metaphor to help us understand. So, and the trees always go usually they either go left to right or bottom to top. So, time here goes from, past to present, or the other one, it goes left to right. And I'm never quite sure why in most of the sort of evolution of human time life scenario layouts, you always see time always goes from left to right. I'm not sure what in the Western tradition why that is. Maybe I don't know, but the idea is to convey some kind of transfer of time and things changing over time.
Right. Oh, and in the West, words on a page go from left to right also.
That's true.
So we have that convention built in.
Yeah. So Darwin initially, the little dots connecting things here were meant to show relatedness, and then relatedness and change over time, and that kind of changed it from that Aristotelian ladder of progress to something a little more detailed. But implied in that, of course, is that they're branched off, they're broken off,
and moving outward from trunks, or a single trunk that was sort of in the center of the page. Yeah, as time progresses, diversification occurs. Species diverge, and eventually you get a great canopy of biological diversity. And that's the idea. Well expressed, well captured by the tree of life, the analogy that Darwin used in The Origin of Species, where he says, it seems to me that life is like a great tree. I haven't memorized that quote, but it's in the book. And so that was Darwin's simile. The phrase tree of life was much older, of course. Went back to the book of Revelations, chapter 22, and it was essentially a sort of a vague religious idea, the tree of life, a wellspring of blessings from Christ or whatever. And then, really, with Darwin, it took on a new meaning. It took on an evolutionary meaning, and that was descent with modification from an original single or very few primitive life forms through time on the vertical axis diverging on the horizontal axis to create different limbs of and branches of biological diversity. So he concretized that. He solidified that as the image of evolutionary history.
And it works pretty well for helping us grab the bigger picture.
Yes, yeah. And even people who are involved in this molecular evolution, revolution that I describe in the book, many of them will say, as a first take generalization, the tree is a good model, except there are aspects we now recognize, aspects that were invisible before genome sequencing, before we could compare genomes letter by letter. There are things that occurred that don't conform to the tree model. And you mentioned earlier, more like a web or a net. Although a web and a net, those aren't the ideal metaphors either. It's the discoveries that I describe in the book are so counterintuitive and so complicated that there isn't a neat single visual metaphor that captures it.
Yeah. One of the things I really appreciate about your writing, in addition to being a really good writer and narrative storyteller, is that you nicely encapsulate what the book is about at the beginning for us who don't know much about this particular topic, which is most readers. By the way, the book is really beautifully produced. Simon & Schuster does a nice artistic design of the cover and the layout, the typography, and all that. I'm one of those older dudes that still appreciates physical books.
And I'm very, very happy with what Simon & Schuster did.
Yeah, I read newspapers. I like audiobooks, but I also like physical books. So you talk about the three surprises that kind of launched this whole field of molecular phylogenetics. So phylogenetics, like the phylogenetic tree, is the evolutionary tree at the molecular level, is the description here. One of these three surprises involves an anomalous form of creature, a whole category of life previously unsuspected and now known as the archaea. Their name gets uppercased when used as a formal taxonomic category. Another is a mode of hereditary change that was also unsuspected, now called horizontal gene transfer, HGT, horizontal gene transfer. The third is a revelation, or anyway, a strong likelihood about our own deep ancestry. We ourselves, we humans, probably came from creatures that as recently as 40 years ago were unknown to exist. And then you say, taken together, these three surprises raise deep new uncertainties and carry big implications about human identity, human individuality, human health. We are not precisely who we thought we were. We are composite creatures, and our ancestry seems to arise from a dark zone of the living world, a group of creatures about which science, until recent decades, was ignorant. Evolution is trickier, far more intricate than we had realized. The tree of life is more tangled. Genes don't move just vertically. They can also pass laterally across species boundaries, across wider gaps, even between different kingdoms of life, and some have come sideways into our own lineage, the primate lineage, from unsuspected non-primate sources. It's the genetic equivalent of a blood transfusion, or, different metaphor preferred by some scientists, an infection that transforms identity. Infective hereditary. So that's really nicely said. And that really encapsulates this whole new area that most people, I think, don't get. You don't see a lot of Nova shows that are on evolution delving into this, and this science is all within our lifetime.
It is, and it began, the starting point, in a way, was November 3rd of 1977. I try and lay out the big intellectual surprises in the foreword to the book, as you say, and thank you for reading that. That's, I hope, will help listeners get a grip on the ideas. But I also want to say that this is a book about the most important biologist of the 20th century that people have never heard of. It's about a man, a very interesting, crotchety, complicated man named Carl Woese, a guy with white hair, a little man, a microbiologist in Urbana, Illinois, at the University of Illinois, who made a discovery and announced it to the world on November 3rd, 1977. Whoa, people, there is a third form of life, a third major form of life on Earth. We thought it was all bacteria and complicated organisms with cell nuclei, eukaryotes, meaning animals, plants, fungi, and everything else. We thought it was bacteria and eukaryotes. And he comes along and he says, there's a third form that it is completely different from either of those two forms, and I have discovered that by looking at fragments of genome sequences, which nobody else was doing in 1977. So what I'm doing in the book, or trying to do, is tell the story of this scientific revolution, but tell it as the story, to a great degree, the story of one man, his peculiarities, his brilliance, his struggle, and his influence on the scientists who have come after him.
Including Lynn Margulis.
Well, Lynn Margulis is an important character in the book, but she falls in a special category. Lynn fell in a special category, as you know, in a lot of ways. The wonderful Lynn Margulis, microbiologist, she was not so much influenced by Woese as promulgating a real revolution that was parallel to his, separate from his. She was saying, beginning in the late 1960s, Lynn Margulis, when she was just finishing graduate school, was saying, look, people, the complex cell, the eukaryotic cell, the cell that has a nucleus with its DNA locked inside the nucleus, the kind of cell that composes all animal bodies, all plant bodies, all complex organisms, that cell has a much different origin story than what we thought. There are organelles, internal organs, like we have a liver and a kidney and a heart. A cell has its organelles, that perform functions. And Lynn Margulis was saying, based on microscopy, based on looking through a microscope, she was saying, look, these cells have been assembled by capturing bacteria hundreds of millions or billions of years ago. And the organelles that our cells contain are captured bacteria. Our mitochondria, this little organelle that's responsible for energy packaging in the cell, is the descendant of a captured bacteria, which is why it has a completely different, now we know, has a completely different genome from the genome in our cell nucleus. In plants, the chloroplast, which does the photosynthesizing, is a captured bacterium. So her theory became known as endosymbiosis, symbiotic relationships of essentially boxes inside boxes, organisms living inside the cells of other organisms. So Lynn Margulis was promulgating that idea in the 60s and 70s. People were saying she's wacky, she's crazy, this idea is nuts. And then Carl Woese and his followers came along and started sequencing genomes to look at relatedness. And people said, well, let's sequence the genomes of what Margulis is talking about. Let's sequence the genome of a eukaryotic cell and also its mitochondria. And they found these genomes are completely different. And the mitochondria genome matches the genome of a particular family of bacteria. So lo and behold, Lynn Margulis was right. She was vindicated for this supposedly crazy theory.
Yeah, it was amazing.
So they converged and overlapped in a sense, although they did not like each other.
Yeah. Yeah, I get that from a lot of the characters you write in the book. It really brings out the human nature of science. It's very competitive, a lot of big egos. Just in the titles of their papers and books, whole new life, whole new species, the origin. They all like to use the origin of species phrase to put in there somewhere to say, I am in the footsteps of Darwin.
Right. Margulis did that. On the Origin of Mitosing Cells was her first big paper. Not a coincidence that it was parallel to On the Origin of Species.
Right. I think most of our listeners will know of mitochondrial DNA because it's always brought up in human evolution stories about tracking our own ancestry through the female line, which carries the mitochondrial DNA, and you can tell relatedness, about how far back we go and so on. But and I was very familiar with that. And then when I read Lynn's work, I thought, yeah, why would mitochondria have DNA? That doesn't even make sense unless it turns out she's right, and it was an independent organism itself, so of course it would have its own DNA.
Right. Yeah, absolutely. And there was a fellow named, it's all about people, characters. Science is a human activity, as you said. There was a fellow named Ford Doolittle, a microbiologist and molecular biologist up at Dalhousie University in Halifax, Nova Scotia, who in the late 1970s, he was one of the very earliest who said, hey, let's test Margulis's endosymbiosis theory by using Carl Woese's methodology and sequencing the DNA in these mitochondria and see what they resemble. And what he found is that they resemble this particular group of bacteria called alpha proteobacteria. That won't be on the quiz at the end of the podcast, people. But he was the one who essentially drove the first nail confirming Margulis's theory with molecular data. Because, of course, I said she was a microscopist. She was a microbiologist of the classic school. She looked through microscopes, and her colleagues created electron micrographs so that she could look at shapes. She didn't do DNA. She was not a molecular biologist. She was a microbiologist. That might sound like a small distinction, but within this field of science, it's a huge distinction.
Mm-hmm.
And so when she, with her microscopy evidence, her visual evidence, was reinforced by the people who were doing the molecular sequencing, the DNA evidence, when she was reinforced by them, then people had to say, well, this is a real thing.
Yeah, a good example of consilience in science, where these different areas of research converge to the same conclusion or reinforce each other, building confidence that the theory is probably true.
Right.
Yeah.
Coincidentally, I just finished rereading Ed Wilson's book,
Consilience, yes, yeah. A classic. Which is William Ewell's idea of consilience. And ironically, he was not an evolutionist. He was skeptical of evolution. So it's because that's pretty much how we know evolution happened, is through this convergence of multiple lines of inquiry independent of one another. Keep pointing to this conclusion. Anyway, so Carl Woese was your entry into this field, and that took you back. It's good to remind ourselves that, in Darwin's day, there was no known mechanism of how heredity happens. And he was something of a Lamarckian, and he had some bizarre ideas about the gemmules doing this and that. And, of course, this is long before sophisticated microscopes would even allow them. So they were – it's a good example in science of a theory that still has a black box built into it. So we know this is happening because we can see it, and there's a black box in there that somehow makes this happen, and we don't know what it is.
That's right.
And, often you think, well, one example I have, like with plate tectonics – in order for continental drift theory to be accepted, there needed to be some mechanism to show what in the world would drive a continent to move. So Alfred Wegener's theories were not accepted until the 60s, when it became clear that there were these cells beneath the continents that were moving and pushing the continents along. Then it became accepted. But Darwin's theory was accepted without that black box mechanism, which really is not until Crick and Watson in 1953. So a lot of your history that you reconstruct shows people trying to figure out what is a gene. I mean, that you have that great line in there about I forget who it was that said, none of us actually know what the gene is. Whoever in his Nobel Prize lecture, I forget who it was. I was just reading this.
I think it was it E.B. Wilson?
Yes, that's right. Yeah. Right, I have it here. It's one of those things. Unless you think about it, it's like, what is a gene? I mean, it's like, what is a species? It's a human convention to describe something in nature. We're trying to get our minds around it, so a gene, we're just talking about something that's happening, but it's not necessarily going to agree on it.
Thomas Hunt Morgan, I think. It might have been he who said that, in a very important geneticist, fruit fly fellow, early 20th century, 1920s, 1930s, and he and the people in his lab were sorting out how Mendelian genetics worked and how they could map different genetic traits on chromosomes, but they were still wondering, is there such a physical thing as a gene, or is a gene more an agglomeration of effects caused by who knows what?
Right.
And they were moving toward that, and then finally they had proof that, well, the gene really is a physical unit, a stretch of nucleotides, of DNA letters assembled in the genome on the chromosome that can change, that replicates, that is passed along in particulate form to offspring. But Darwin, of course, as you say, he knew none of that. It was a black box. Even even Mendelian genetics was a black box to him because people hadn't hadn't rediscovered Mendel until 1900.
Right. I wanted to show some of the other. I mean, you reproduce many of the trees in the metaphors used by them, and I like how you go back in history, like to Ernst Haeckel and his famous trees. I had always used. Let me find it here. This example of. I always liked this tree of Ernst Haeckel, his early tree, which is more like a topiary with richly branching twigs and more like a web or bush. And then later, I use this example, the emergence, the pedigree of man. He had Minchin in the top as an example of his irresistible pull toward anthropocentrism. But you make a good case that actually that was just the pedigree for just humans. So of course you're going to put humans at the top if that's what you're talking about.
Right, and that was a point. I forget which scholar, an academic scholar of Haeckel.
Richards, I think it was Robert Richards.
Yeah, he makes that point. So I owe that insight to him. But I think it's both persuasive and important, so that we don't dismiss Haeckel as being too anthropocentric.
But
Haeckel, yeah, the first great follower of Darwin, who was not only an important biologist but also a talented artist. So he started drawing trees, and his trees were more tree-like, were more vivid, were more artful than anybody else's, and that was one of the reasons that they were persuasive.
Yeah, another one of the great potted histories you put in there about biographies. He had quite the colorful life and lived a long life like Wallace, and so could make a lot of mistakes along the way as well as many great discoveries. That brought up another subject that I'm interested in is the origins of creativity. A lot of the people you discuss, they seem to be kind of quirky, a little off, and a little maybe too open-minded to ideas that are probably not true, but maybe you need to be a little bit like that to bump into one great idea that turns out to be true and is revolutionary.
Well, there are certainly quirky people that have done more than their share contributing. I mean, Lynn Margulis, Carl Woese, absolutely. Ernst Haeckel, absolutely. I don't know whether we should consider Darwin himself quirky or as the most staid of respectable middle-class Englishmen. I say in my little biography of Darwin that here was a fundamentally conservative man who found himself burdened with a deeply radical idea, and he was so honest, so sort of by instinct and personality, such an honest man that he struggled with that with great courage and great candor to sort out that tension between his conservativeness and this radical idea, causing a lot of discomfort to his wife and a lot of his friends, among others.
What is your explanation for his delay of roughly 20 years from when he first developed the ideas about natural selection to 1859 when he finally publishes?
Right. From 1837, 1838 when he sketched the idea in his notebook to 1859 when he published The Origin. Well, it's the famous Darwin's delay. There are a handful or more different theories, and not surprisingly, I think that you can't really say that any of those factors was unimportant. People say, well, he was scared. His wife was very conventionally religious. He didn't want to upset her. He didn't want to get in trouble with the people who had been his teachers and his colleagues who were part of the scientific establishment. Some people say, well, it was the religious establishment of the Church of England in the 19th century. Some people forget that the scientific establishment and the church establishment really were the same establishment in England in the 19th century. A lot of Darwin's teachers at Cambridge had been ordained Anglican clergymen, people like Adam Sedgwick and Henshaw. And then there was the fact that in 1844, the book Vestiges of the Natural History of Creation, which was sort of a Chariots of the Gods slapdash book that contained evolutionary ideas thrown in this great bouillabaisse of facts and semi-facts and distorted facts, and it became a great bestseller. And it was candy. It was candy for the masses. and respectable scientists said this book is junk. This book happens to be promoting the idea of evolution, but it has no rational theory. It's just this potpourri, and it made the idea of evolution unrespectable for a while. So part of the argument is that was published in 1844. Darwin said, "Well, I don't want to come out with my theory right now. People will lump me with this popular book." Plus, he wanted to gather a huge trove of ironclad evidence and argument. Plus, he was sick a lot. I say in my little book that Charles Darwin probably vomited more than any other great scientist in history. He had his mysterious ailment.
Mm-hmm.
And he got distracted for 8 years doing the taxonomy of barnacles. All of those things combined to make him, as the title of my book is The Reluctant Mr. Darwin. He was reluctant to publish this theory until the time was completely right. And then our hero Wallace came along and jabbed him,
right,
scared the bejesus out of him that he was going to be scooped after all these years. And so he rushed ahead and wrote the quick and dirty version of his long book, and the quick and dirty version is what we know as The Origin of Species.
Right. Yeah, that's how I always read it too. I think he got one of his friends, I think it was a botanist, might have been Henslow, or I forget who it was, that wrote him about the vestiges of creation and said, don't do this. You need to spend time piling up a lot of data and information and evidence. Oh, Hooker. It was Hooker. Yes. Or else you're going to look like this guy, unprofessional, and so on.
I think Hooker said something, and then I think it was Darwin's reply saying that no man should describe the origin of species who hasn't painstakingly described many species, described in the taxonomic sense. So that was one of the reasons he spent 8 years on the taxonomy of barnacles, because
right.
he was proving that he was very capable as a technical taxonomist to say, here's where one population of animals ends, that's a species, here's where another
right.
Connected population begins. He did that with barnacles and made his bones as someone who was entitled to talk about, okay, now we can discuss the origins of species.
So you can see why most of your book takes place in the 20th century, particularly the second half of the 20th century, because if it's so hard to figure out these relationships with big things you can see just by walking outside, how much harder is it for things you can't even see with your naked eye? You need the technology to be able to do it.
Right. And Carl Woese began with microbes. He began with bacteria and this third form of life that he discovered, the archaea, discovered that they were not in fact bacteria. They look like bacteria through a microscope, but they're more closely related to us at the molecular level than they are to bacteria. So he started at that level because they're relatively simple organisms with relatively small genomes. And therefore, he could, with his painstaking 1970s methodology, he could sequence fragments of their DNA, just fragments, not whole genomes, because it was so difficult. It involved explosive chemicals and toxic chemicals and radioactive chemicals and high voltage. And he jerry-rigged this apparatus in this method where he and his assistants, his graduate students and others could extract bits of, actually they were working with RNA and not DNA, first of all, bits of RNA, and they were complicated but good reasons for that, from these microbial cells, and then break those stretches of RNA into fragments, and then sequence the fragments, maybe just 4 or 5 letters long, A, G, U, U, C, G, C, A, A, U, things like that. And he sequenced these fragments, and then he would compile, he called it a catalog, but essentially it was a paragraph of words. The words were these DNA fragments, and the paragraph characterized a particular species. And then he would compare one paragraph of these RNA fragments to another, and he could see how closely things were related. And then gradually, as gene sequencing methodology and technology increased, and computing capacity increased during the 1980s and the 1990s, fairly soon, was a trivial task, relatively trivial, to sequence an entire genome. I mean, the human genome was done in 2000, 2001. That was the first big, complex, complete genome. There were some bacterial and some archaeal genomes sequenced before that. But as a result of this, by about 2004, 2005, there were complete genomes that were publicly available online, and people could download these and compare them. So there was an explosion of understanding of how genes had moved around from one genome to another. As people could screen these genomes, look for strange anomalies, and they started saying, "Wait a minute, here's the genome of an opossum from South America, but it's got this stretch of DNA in it that comes from a kissing bug. How did that happen? How did the kissing bug DNA get into the possum?" And they made a number of discoveries like that that illuminated the fact that this horizontal gene transfer phenomenon has been happening, and it's been consequential.
It is almost unfathomable. I could, on a macro scale, like a possum or a human. So let's start at a smaller scale. How would, say, whatever the mitochondria was when it was living on its own, and it gets incorporated symbiotically into another organism, how does it then continue that process? How does the replication of the mitochondria inside a cell carry forward genetically or reproductively?
Well, I don't think anybody knows exactly how the captured bacteria in those early cells, first of all, how they got in. Did they get in because a bacterial cell swallowed another bacterium? That's possible. Or did it get in because one bacterium infected another bacterium? That's possible too. One of the amazing things is that we know from looking at the DNA that it only happened once in the history of life. The mitochondria in complex creatures were acquired once as captured bacteria, one event, at least one event that left living offspring. So sometime 3 billion years ago, 2.5 billion years ago, a single bacterium got into another bacterium and managed not to be digested, managed instead to replicate itself. So now there are thousands of mitochondria inside that particular cell, and when that cell reproduces itself, it divides in half. Both of those new cells have a bunch of mitochondria in them, and each of those mitochondria carries a unique genome, different from the host cell's genome, and each of them is capable of replicating itself abundantly, so that I describe it in the book as the mitochondria in a cell are like flakes of pepper in a stew.
But replicating. So you split, you divide the soup up into two bowls, and they keep replicating. So the ratio of peppercorn in the soup stays the same.
Right.
Yeah.
You add more tomato sauce and put it out into bowls, and it
keeps replicating. Yeah. So to me, so presumably then they have a reproductive advantage or survival advantage over cells that don't have mitochondria. So then, therefore, they survive and succeed, and that's Darwinian.
For starters, they have these mitochondria that package adenosine triphosphate (ATP), this molecular form of biological energy. They package it into these units like AA batteries that are useful to the cell. So now the cell can do things that a bacterial cell cannot do. And there's a great body of thought, very lively right now, as to exactly how this happened and whether complex eukaryotic cells became complex and then acquired mitochondria, or became complex because they had acquired mitochondria. There are two very emphatically disagreeing schools of thought on that.
But just the very idea that gives a survival advantage is a Darwinian idea.
It is absolutely. So Darwin doesn't disappear from this. And Darwin doesn't disappear because of horizontal gene transfer either. One of the assumptions that comes down with 20th century neo-Darwinism is that um, uh, evolution occurs by natural selection acting on variation within genomes. Individuals differ from one another in their characteristics, and those differences are uh, are important in survival and reproduction. So there is the survival of the fittest. Natural selection works on variation within populations. It was thought that that variation came from itty-bitty point mutations in DNA, incremental mutations that added up as they were selected into major adaptations. And that's still true. But we now know also that another form of variation upon which natural selection acts is that sometimes genes come flying in intact from a completely different species, from a completely different kingdom of life, and that brings innovation and makes possible new adaptations. It opens new possibilities for natural selection to shape into adaptations that allow creatures to survive, reproduce, and prosper.
Maybe we should give our listeners a big picture for just a moment of the different kingdoms. So you have this graph in there of the bacteria, eukarya, and archaea. So these are three different. This is 1999, so this is not that long ago. This is Doolittle's reticulated tree.
Right, that's one of the tangled trees.
Yeah, tangled tree. So there you see, this is the horizontal gene transfer going on here, right? Because they're infecting each other.
That's right. Yeah. So that was in the midst of a ferment of thinking, 1999, 2000, up until Darwin's 200th birthday in 2009. People were starting to say, wait a minute, the tree of life is not a tree. The tree of life is tangled. Maybe it's sort of like a net, sort of like a web. It's got these branches, as Ford Doolittle's drawing of the tree shows. Some branches go sideways and fuse into other branches. That's the horizontal gene transfer. So this is completely different from what we thought, or radically different from what we thought. Not completely, because the background shapes are still the tree, and yet there are limbs doing things that limbs on a living tree never do.
Mm-hmm.
And it came to the point where in 2009, you may remember this, Michael, the popular science magazine New Scientist.
Mm-hmm. I do remember this.
In observance of Darwin's 200th birthday, they put a tangled tree like that on their cover, and they had a big headline that said, "Cutting down the tree of life," and inside articles with titles like, Darwin was wrong.
Yeah.
And what they were getting at was that the story was much more complicated. It wasn't just incremental mutation that brought variation within lineages that always diverged. It was also horizontal gene transfer and the tangled tree of life. But of course, when they had a headline saying Darwin was wrong, creationists jumped on that and said, "Hey, see, these guys are finally admitting it." And some of our friends and colleagues in the scientific world, like Richard Dawkins, like Daniel Dennett, like Jerry Coyne, they went ballistic at this, and they wrote to New Scientists and said, "What are you doing?
Right.
In comfort to the enemy,
right.
You're confusing everything by overstating the degree to which horizontal gene transfer and the new tree of life challenges Darwin."
And it is a little bit of an overstatement because it isn't all web.
Right.
The tree metaphor still works for most things. I mean, for us, for example, you talk toward the end of your book about, Human Genome has about 8% of foreign genes in it. Something like that, right?
8% viral.
Viral, sorry. Yeah, viral. Yeah.
Human genome is 8% viral. Should we talk about that?
Yeah.
That people say, wait a minute, what are you talking about? That's counterintuitive. Well, one of the things that we have learned through this sequencing of complete genomes and comparing them with high-powered computers is that 8% of the human genome, yours, mine, and everybody else's, is composed of viral DNA that has been captured in the course of millions of years, but captured sideways and not descended vertically through the entire ancestry of animals. We have DNA, we even have functioning genes that came into the primate line by infective heredity. Retroviruses, which copy themselves backwards into cell nuclei, which is why they call them retroviruses. Retroviruses infecting not just immune cells, the way HIV does, but infecting germline cells, egg cells, sperm cells, testes and ovary cells. And when a virus does that, a retrovirus does that, and inserts itself into the genome of one of those cells, it becomes heritable. It gets passed down to offspring. And so this is accumulated. So we now have 8% of our genome consists of captured retroviral DNA.
So this must have happened, if everyone on Earth has it, then this must have happened something like 100,000, 150,000 years ago or earlier.
Earlier. I mean, it's happened a number of times. At the end of the book, I described the work of a Frenchman named Thierry Heidemann. Fascinating, important scientist. I flew to Paris just to see him. I emailed him and said, will you see me for an hour if I come to Paris? And he said, yes, I will. And he was very accommodating. I ended up talking to him for 7 hours that day. And it made the trip worthwhile, telling me about his work. And his group, they have discovered, in particular, one gene that's called syncytin, Syncytin, syncytin number 2. And it is a gene that was captured tens of millions of years ago within probably the primate or the pre-primate lineage, captured by infection with a retrovirus that got into the germline, got into the reproductive cells. And this particular gene was originally a gene that in the retrovirus made the viral envelope, sort of the membrane around the viral capsule. Do you have a little bit of air?
Yeah, I have. Let me close the door here. Hang on.
Oh, okay.
Weed whackers and tree trimmers.
Okay. I thought it was some sort of an electronic interference on our line. It was just
it was the outdoor.
Okay.
Yeah, no. So
Thierry Heidemann and his group discovered this gene, syncytin 2, that originally was an envelope gene in its retrovirus, and got into the primate line, including the human line, and it was adapted, modified, so that now it creates a very different kind of envelope, a very different kind of membrane. It's the membrane between the placenta and the fetus in a pregnant female.
Right.
And without that membrane that's created by a viral gene, pregnancy cannot be successful.
For all mammals.
For all mammals. But it's happened a number of times in the mammal lineage, and it seems as though, I asked Thierry Heidemann about this, well, I mean, how can you be a mammal if you don't have this crucial membrane? And he said, well, syncytin 2 is a late acquisition, relatively late, and presumably in pre-mammalian animals, a gene like this was captured, a viral gene, and originally it had some sort of a function. It was adapted to some sort of a function, an immune function of protecting the creature against other infection. And that immune function then was adapted over time to the possibility of having internal pregnancy without an eggshell. And because you could create this membrane, and it would make the fetus immune to the protect the fetus from the immune system of the mother, because of course the mother has a different genome than the fetus, and so her body would be inclined to reject that. That's why birds lay eggs, is because they put a shell around it. It protects the chick from the immune system of the mother, and then they lay the egg, and then they sit on it and protect it. But there was an advantage in internal pregnancy. Evolution found that advantage, made it possible with this membrane that protects the fetus from the mother's immune system, and also now incidentally carries waste products away from the fetus so that the mother can void them out, and carries nutrients from the mother across this membrane into the fetus to feed that fetus. So that fetus, instead of having a yolk in an egg,
Mm-hmm.
it has a continuous flow of nutrients from the mother. All of that is done by this membrane that is coded for by a gene that has gotten into us by viral infection.
So if this is true for all mammals, then that would put it back to 60, 70 million years or more.
Exactly. Yes.
Wow.
Yeah. That's when the first syncytin genes
Yeah.
were being created in the animal lineage.
And this would be an example of what did Gould call that? A co-option. It was co-opted. It was, in other words, a feature that originally evolved for one purpose. In this case, didn't evolve really. It's just lateral gene transfer, and then it becomes adaptive for some other reason.
Right. And Thierry Heidemann would say, this is our hypothesis, and we're investigating. We're trying to test that hypothesis that first of all, it performed an immune function, and then it was co-opted.
Yeah.
Also, this intermediation of waste products and nutrients between the mother and the fetus.
Yeah. And just to punctuate how counterintuitive and radically different this isn't like we all have 2.5% Neanderthal genome in us because 150,000 years ago or 50,000 years ago or whatever on a cold cave night, a Homo sapiens and a Neanderthal, got together. This isn't like that at all. This is something completely different.
Right. That's the hybridism that we were talking about when we were talking about Ernst Mayr.
Yeah.
Two closely related species can sometimes, interbreed and produce offspring.
Right.
So we have Neanderthal genes because of that. This is different. This is like if Homo sapiens interbred with a bacterium or interbred.
And this could have just been some just random infection.
Yes, I mean it presumably was a random infection. We're constantly being bombarded by viral infections.
Mm-hmm.
My most recent previous book was Spillover. Was all about zoonotic diseases, viral infections that come to humans from animals. Ebola, Nipah virus,
mm-hmm.
Hendra, Zika virus, West Nile, and all that, and HIV, which came to us originally from chimpanzees. So I forget what point I was making with that.
Oh, well, we were talking about how different this is than sexual reproduction and hybridization. So how confident are the scientists that do this that the 8% figure is about right for what we have in our genome?
They are very confident of that.
I mean, it could be higher, even more.
The human genome has been carefully looked at.
Yeah.
High-powered, computation tool.
Yeah.
It's another interesting story that I tell in the book. When the human genome was first published, Craig Venter did a rough draft of it as a private enterprise in 2000. Mm-hmm. The Human Genome International Consortium Project produced its in 2001. And when they published their draft of the human genome, the whole group of the consortiums in the paper said, well, it's very interesting. One of the things they said is very interesting. There seem to be 223 cases of horizontal gene transfer from bacteria into the human genome. And people said, whoa, wait a minute.
What?
223 cases where bacterial genes have gotten into the human genome? And then people examined that claim very closely, and they walked that back. That was not correct because they didn't have enough other genomes for comparison at that point, and some of that was a matter of a complicated matter of gene loss in some species rather than these things having come directly from bacteria. Mm-hmm. But that was one of the first trumpet blasts saying, look, people, you've got to take horizontal gene transfer very seriously. That was an overstatement, an overclaim, but for the following 17 years, we continue to find more cases of horizontal gene transfer in not just at the microbial level, but among complicated organisms like animals, even humans.
So something like GMOs, the complaint by the anti-GMO people is that this is not like breeding corn where you just keep breeding them and make them bigger. You're actually sticking new genomic sequences into a tomato or a fish or whatever from a completely different species. essentially this is what nature's doing, or CRISPR that you talk about in your book would be what we're doing artificially, nature's been doing for billions of years.
That's right. And I try and make that point. I mean, it's not a book meant to argue that, germline genetic engineering is a good idea or is always safe. But I try and make the point that if people are saying, hey, don't do that, don't take a gene out of a jellyfish and put it into a tomato, that's not natural. Well, in fact, it is natural. It's been going on for 3.5 billion years.
Yeah, this is quite striking. So I guess the question is, to what extent does this modify the traditional Darwinian view of evolution? 8% different, 50% different. To what extent do we have to reconsider explanations for the origins of things like wings or fur or feathers or whatever?
Well, I think where it really changes things is our understanding of the source of novelty, the source of variation. And as I said, the conventional neo-Darwinian view is that, natura non facit saltum. Nature does not make leaps. Darwin himself embraced that axiom. Turns out that axiom is wrong. Nature does make leaps. Form of horizontal.
Sometimes.
Right.
Yeah.
Right. Yeah, absolutely. Sometimes. And so one of the sources of variation on which natural selection acts is incremental point mutation, the old-fashioned way. Another important source of variation, in some, perhaps many cases, is horizontal gene transfer, when whole genes and package of genes come into a lineage suddenly, and then natural selection has the opportunity of either closing off that lineage or taking it into interesting new possibilities.
Yeah, your discussion of the placenta makes me wonder what other major features that have long puzzled scientists, like the origins of the wing or fur, feathers, or whatever. You can certainly see the adaptive advantage once you have a full structure, aerodynamic structure, or whatever, that's doing what it's doing now. But how did it get there at the halfway point? 50% of a wing is useless as an aerodynamic structure. The problem of incipient stages. Right. Maybe this is going to be an area of research going forward to explain some of these macro changes that we see that are difficult to see how they could have gotten started in the first place and then built up to a full structure.
That's an important point, and I'm sure people are going to be asking those questions and querying genomes to try and find answers to those questions. I can't predict what the answers are going to be, but it's a very interesting line of inquiry.
I also like how you reflect toward the end of the book, kind of philosophically, on the nature of the self. I was just writing about this. My latest book is Heavens on Earth, about the search for the afterlife, and this whole idea of the soul, and that either in the religious version where you're replicated in heaven with Jesus and God somehow pulls you out of the grave, or it's just a soul, or you go the Ray Kurzweil singularity. We're going to copy your connectome and put it in a file in the cloud and turn it on, and you're going to be there. This is all under this assumption that you're just a snapshot of you at this very moment, and the copy of your genome and your connectome. That's it. But really, we're this constantly changing, fluid set of things. Of course, I go through all the stuff that you aren't the same person you were 10 years ago. All the atoms in your body have been recycled, and the cells have all died. And these are new cells made out of new material. It's the pattern. You talk about Theseus' ship in the book. It's such a great story. So who are you? You're this pattern, but the pattern itself changes. And the things you're talking about in this book that makes this problem even worse. We're not even just primates. We're also viral, and not just the bugs living in your gut to process food, in your cells.
In your cells, in your DNA, we are mosaics. Yeah, we are composite creatures, and the idea of an individual, including a human individual, is challenged in interesting ways and requires modification because of the discoveries of Carl Woese and his followers. What is a human individual? If there are descendants of bacteria in every cell in the form of mitochondria, if 8% of the genome is captured viral DNA. What does it mean to say, I am a human individual, you are a human individual? Now we realize that we are composite organisms. We're mosaics. What that does to identity is tricky enough to contemplate. What it does to religious belief,
right?
That's your
yeah. Well, it seems to me this strengthens even more this kind of reflective idea that we're all connected. All life forms are connected to each other in this long chain going back, 4 billion years or so, and even more interconnected than we ever imagined.
More interconnected and intertangled.
Yeah. Intertangled. That's the right word. Intertangled. Yeah. Yeah. Those tangled webs.
Right. Yeah. And so we can't say, well, here we are, to go back to Ernst Haeckel and his tree of man. Here we are at the top of this great tree, the pinnacle of creation, and way, way down at the bottom are things like bacteria and, pondweed. And on a dotted line going way out to the right is this group called viruses. We don't even know exactly whether we should consider them life or some sort of mechanistic other thing. And now we realize, well, there are strong connections directly between us and the viruses and directly between us and the bacteria. They are part of who we are.
Right. Yeah, treating them as if they're down there and we're up here in this exalted state is incorrect. Are you familiar with Paul Davies' idea of a second Genesis? Paul Davies is one of the SETI scientists looking for extraterrestrial intelligence, and he has this idea, we don't have to go to Mars, we can just search right here on Earth. If you found something that had a different kind of self-replicating molecules, instead of DNA, it was something completely different. That might be a second Genesis on Earth that might have happened.
I'm not familiar with him, but I get that idea.
Yeah.
I mean, that would be as extraordinary as that would be more extraordinary than finding bacteria on Mars.
Yeah, it would. Yeah.
That's sort of what Carl Woese's discovery of the archaea did, but not so extreme, because the archaea, completely different from bacteria, completely different from everything else, but they still use the universal code.
Right.
They still use the same DNA code. A certain combination of DNA letters still makes that amino acid and no other. Well, those several amino acids. So it's the important evidence of the universality of the DNA code. And what Paul Davies is presumably talking about is, what if we found a group of organisms that used a completely different code, a completely different language? That would be big. That would be.
Yeah. Also, reading your book, it made me appreciate how difficult the problem of the origins of life research is. I mean, that's even going back even further from where you start. There's already self-replicating molecules and organelles and things like that. The harder problem is even, where did even that come from? Like like cell wall, protein-based cell walls. Why would that have formed? How did that form?
Right. One of the things that was a revelation to me in researching this was this concept called the RNA world. Back before the first evidence of cellular life on planet Earth, before 3.8 billion, before the earliest protobacterium, what was life then? And Walter Gilbert and some other people speculated in the 1980s, including Woese, that the starting point was RNA, not DNA, which is a complex, stable molecule, double helix linked together and everything. Some people say, well, it's the boring molecule. And RNA is the really interesting, changeable, plastic molecule. So they talk about the RNA world. Maybe the first forms of pre-life were RNA molecules that were competing for the components, the nucleotides, that one particular RNA molecule might be competing against another RNA molecule to build itself longer. No cell wall yet.
Right.
No containment, just these naked molecules in the in the primordial soup.
In the soup, or in the ice, or whatever the medium, the mud, something that was holding it together.
On the surface of the moist rock.
So if we found bacterial grade, or even before that, just if we found something like RNA on another planet, but not yet DNA, or both RNA and DNA, that would be a sign that that's a natural process to go from RNA to DNA. Something like that. There was a guy, it was Lynn Margulis' first husband, I forget his name, who was interested in this subject. Oh, yeah, Carl Sagan.
Exobiology.
Yeah, exobiology.
Yeah. And then she was interested in endobiology.
Right. Remember Ed Wilson told me a funny story once how when he was young, he'd just gotten hired at Harvard, and Sagan was at Harvard, and came to him one day to ask him about the ecology of Mars. And he's like, the ecology of Mars? What ecology? What are you talking about? Well, let's just speculate. Oh, okay, here we go. That was the young Carl Sagan. Let's imagine how that goes. Well, that's how it starts with these, asking the deep questions, the origins of life. How did these things ever get started?
And actually, Dr. Carl Woese, he got an important component of his early funding from NASA.
Oh, right.
As part of their exobiology project, because he was dealing with these fundamental questions of, the first question for him was, how did the genetic code itself evolve? How did that come to be, the genetic code? These constituent letters produce these amino acids to be linked together into these proteins. How did that start? NASA gave him money to think about that and to work on that because they thought it might have something to do with ecology of Mars.
Mm-hmm. Interesting that as you write about him being kind of stuck in this small town in Illinois and not a great communicator, and not a good speaker, teacher, and all those things end up factoring in to what extent your ideas get out there, or not.
Right.
And you have someone like Carl, who was a great communicator, or Lynn Margulis, who was also she was out there kind of pushing. She was had a forceful personality to be able to to get her ideas out there. Sometimes that's what it takes.
Right. And he did not have that, and that's probably one of the reasons why he is the most important biologist of the 20th century that most of you have never heard of.
Right. Well, thanks to you, you're resurrecting him.
I hope so.
So where do you see this research going, say, in the next 50 years or so? I mean, you must have had some idea of the trends that they're pushing toward.
Well, it's so nonlinear. Such an explosion of possibilities that obviously there's going to be a lot of push using CRISPR and other technologies to decide whether we want to reshape our human babies and other creatures on Earth using the tools that we have. And I think one of the things, I said that this work challenges the categorical idea of an individual, the categorical idea of the species. Those challenges are just going to become more and more severe and more confusing to us as we balance between what we can do and what we should do.
The transhumanists I dealt with in Heavens on Earth, they're all for it. Just go for it. Let's just completely modify humans. We'll be post-human. Yeah. When we colonize Mars, we're going to have a different species. We'll become a different species, but not evolutionary-wise, but through CRISPR technologies. We'll just make it happen. Problem, it seems to me, is that it's always more complex than you think it's going to be, and there are going to be unforeseen circumstances or not predicted. I'm sorry, I'm spacing out on the phrase. The unintended consequences.
Yeah, unintended consequences. Right. Yeah, I completely agree with that. I think there are some other issues too, but I completely agree with that. It's so difficult to predict what the consequences. It's difficult to create a climate model of the global climate and predict 1 year ahead what's going to happen. How difficult to predict once you have an unlimited capacity to move genes around from one species to another by technological means. What's going to happen? Not just what's going to happen to the concept and the identity of a human being, but what's going to happen to biological diversity? What's going to happen to grizzly bears? What's going to happen to lowland gorillas? What's going to happen to polar bears? Right. Are we going to one of the that I make, the little stories in my book, is that some scientists argue that there is no such thing as a species of bacteria because they exchange genes sideways so readily by horizontal gene transfer. We now know, and there's a whole bit on the transfer of antibiotic resistance from one kind of bacteria to another, which is what's making that problem so difficult. But these scientists argued that there is just a bacterial superorganism. All bacteria on planet Earth are potentially exchanging genes with one another, and it makes no sense to call them different species. It's just the bacterial superorganism. Well, our technology is going to make possible the approach to that state with all species on planet Earth. I mean, we could, not just moving one jellyfish gene into a tomato to give it better frost protection or something, just moving genes around at will. What's that going to do to wildlife? What's that going to do to our systems? What's that going to do to the great animals and plants, sequoia trees, Bengal tigers, African lions, that are important to human sense of wonder and human imagination? What's going to happen? I don't know.
I know it's huge. Our mutual friend Jack Horner, he wants to bring back, the dinosaurs through chickens or what. I forget what he's working on now. Right, yeah. And which would be just totally fascinating, or maybe more modestly, just bring back the woolly mammoths using elephants or something like that. But even that, well, where do you put them? Where are they going to live? Northern Canada or something, or Siberia.
Their ecosystem consisted partly the ecosystem of a woolly mammoth consisted not just of trees and rocks and ice, but all of the other creatures that they interacted with, and some maybe many of those creatures are gone now.
Yeah.
If you brought back Babe Ruth and put him at home plate, if you brought back who's one of the great pitchers, gone to his reward. Honus Wagner, was he a pitcher?
No, he was a hitter, but what's it? One of those. Oh, well, Cy Young, he was one of the early pitchers.
So you bring Cy Young back and put him on the mound. But where's his team?
Yeah,
that's not a very good analogy, but you see what I'm saying.
Yeah.
You can do a lot of things, but you can't turn the clock back. You can maybe somehow produce a pseudo mammoth from found DNA and living elephant eggs.
Right.
But you can't produce a genetically viable population of mammoths, and you can't produce a mammoth ecosystem. So why spend all of that time and money trying to approximate that accomplishment when we've got all these thousands and millions of other species that still live on planet Earth that are going extinct? Why don't we worry about saving them? That's my feeling on that particular question.
Isn't J. Craig Venter, doesn't he have a gene storage program going on where he's capturing the genomes of species that are endangered in case they go extinct?
Wouldn't be surprised if his institute, Tiger, the Institute for Genomic Research, is doing something like that. I think there are other efforts in that vein. Not only seed storage, but cell, cryogenic cell preservation of the DNA that's inside of it. I think the San Diego Zoo even does that. And there are some valid conservation applications for that. I've written about that kind of thing in pieces for Harper's. But to bring back species, the passenger pigeon, Tasmanian tiger, the woolly mammoth, it seems to me misguided biotechnological showboating.
Yeah, let's save the ones we have before they go extinct.
That's my feeling.
Yeah. Remember when I first met Jack, Jack Horner, he was challenging the very idea of the concept of species, mainly because so many of these words and labels we use are human conventions that confine the way you think about organisms, and really that's kind of what your book is about, is people that broke through that convention and said, let's think beyond that box of species and how they change.
Right. Yeah, and I agree with Jack on that. I mean, we know it's more clearly that the other categories of life into which we humans have sorted and classified, like genus, family, those are artificial human constructs. But at base, even under Ernst Mayr's definition, the species is an artificial human construct, because creatures are doing whatever they have to do and whatever they can do. Sometimes there's hybridizing, sometimes there's horizontal gene transfer, genes moving backwards among bacteria. Species, I agree with Jack. Species is a human construct. I think it's a very, very useful human construct. We shouldn't forget that it is a human construct.
One of the more striking things Richard Dawkins ever wrote that made me sit up, among the many things, was that no Australopithecine mother ever gave birth to a Homo child.
Yeah.
And it's like, right, yes, of course they didn't. So how did we end up with Australopithecines and Homo erectus and Neanderthals and so on?
Yeah. Because they're big gray areas.
Right.
In fact, the fact that black is different from white doesn't mean that there are no gray areas.
Yeah, that's right. Yeah.
With different zones in between there. Yeah.
Right.
And I think that's a humbling message, that nature still in 2018 is more complicated and more various and more intertangled than we can get a grip on with our scientific instruments and investigations and language.
Right. Well, David, we've been going almost an hour and a half here. What's next on your agenda of big books or articles for Nat Geo?
Articles for Nat Geo, that's easier to answer. I'm not talking about yet because I'm not exactly sure what my next book project is, although I have a couple of tantalizing ideas. Meanwhile, I do a lot of work for National Geographic. Right now, I'm on a multi-part project called The Last Wild Places. I'll be doing a series of articles for National Geographic magazine on some of the last wild places on planet Earth and the ways that particular conservation organizations and conservation models are bringing new ideas and new hope in the 21st century to succeed in ways that the old methods and the old ideas haven't succeeded so well so far. So I've just been in Mozambique for a period of time to do a story there on a wonderful project called the Gorongosa National Park Project. I'll be doing a story on the American Prairie Reserve, which is here in Montana. And I'll be doing a number of these over the next 2 years or a little bit more. They'll appear in National Geographic magazine, and then they will appear in expanded form as a book from National Geographic Books.
Is one of these places, I think it was Ed Wilson who wrote about the DMZ, the demilitarized zone, is a place where nature has come roaring back and it's kind of natural?
That's I've heard about that. I've never been there, but that's not on this particular menu of places, the DMZ.
It might be safe to go at the very moment, but maybe not for long.
I'd take off for the DMZ tomorrow to see. I've heard too that I think they have populations of leopards wandering around in there and animals, because it's an irony that military preparedness and military installations and military tensions even sometimes are horrible for habitat and wildlife, and sometimes are good for habitat and wildlife because they keep other human activities out.
Yep.
White Sands Missile Range is a wonderful ecosystem.
Right. No one wants to go there. That's right. Well, David, thank you so much, and congratulations on your beautiful book, The Tangled Tree, A Radical New History of Life. It's a great read, really. You're such a beautiful writer. I really enjoyed reading it. So thanks for coming on the show.
Thank you. I've really enjoyed talking to you. Great conversation. Thanks for having me.
We'll talk to you later. Bye-bye.
Bye-bye.